I don't wanna grow up, I'm a curious kid. The world has 100 questions I can play with. So I'll open my arms and eyes and wonder every day till the day I die. No one really knows why gravity exists.
All right, here we go. Here comes our first speaker. UC Berkeley plant biologist, Chandler Sutherland, on It's Not Easy Staying Green, Understanding Plant Immune Systems. As an undergrad, Chandler studied chemistry and molecular biology at UC Berkeley. And now at Cal, as a PhD student, Chandler does research with Professor Chris Niyogi to understand photosynthetic energy conversion, photosynthesis in plants and algae. And Chandler just does some serious science outreach with Cal's CLEAR program. CLEAR stands for Communication, Leadership, and Education in Agricultural Research. This woman knows agriculture. Please welcome UC Berkeley plant biologist and Wonderfest Science Envoy, Chandler Sutherland.
Thank you, Tucker, for the very warm introduction.
There we go, sharing some screen.
Wonderful. Okay, if you can all see my screen, I will instead invite you to look at my camera for a moment. So this is a fiddle leaf fig leaf. I found it walking around my neighborhood. It had been abandoned, and I'm sure you can guess why. It's looking pretty crusty and dead. If you're a Bay Area gardener or a houseplant enthusiast at all, I'm sure you know that there are a million ways that plants can die and wither and be a little disappointing. They can have too much water, too little water, too much sun, too little sun, all common culprits. But the issue here with this fiddle leaf fig and what we're here to talk about today is disease. Plants like us face a barrage of disease attacks every day. And it's a lot of the same things that try to infect us. Viruses, fungi, bacteria, they all bother them too. And they have the extra effort of dealing with herbivores and nematodes and things that are trying to constantly eat them that we don't have to deal with. Plant disease is important to us because of our reliance on plants for food. 40% of global crop production is lost to disease and pests, and plant disease can be severely detrimental to human quality of life. One of the most famous examples was the Irish potato famine, which was a result of a fungus-like plant disease. Here on the upper left corner, I'm showing rice blast, which is one of the most prevalent plant diseases today. And then here's an example of the potato fungus. Like many things, plant diseases will only worsen with climate change, but I'm not here to tell you a sob story. Hope is not lost. Plants are marvelous at self-defense and are incredibly successful with a set of tools that are very different from our own when it comes to fighting disease. By studying and understanding plants' innate defenses, we can better protect our favorite edible plants and understand and appreciate our own disease-fighting tools. Today, I'm going to be guiding you through a very silly medieval-style battle between pathogens and plants, but I want to acknowledge the very real and devastating ongoing war in Ukraine. I want to say that I stand in support of Ukraine and the Ukrainian people, and that this war is destroying homes, communities, and taking lives, as I'm sure you're all very aware, and has already had a very large impact on our food system. A quarter of the world's wheat is exported from Russia and Ukraine, and as I'm talking about how plants are subject to disease, I think it's important to remember that food systems can also be very human. And that at the end of the day, the goal is feeding people and preventing kind of humanitarian crises. So with that brief caveat, I plan for the next 20 minutes to guide you through the plant immune system and build on the understanding of what plant immunity is, exploring 3 layers of defense plants have against invaders from the perspective of a successful disease. Then we'll turn our focus to how new tools are forged in this fight. How do plants evolve to recognize new diseases and evolve new defenses? And what does the future of this battle hold? Any good story has characters. And so I'm introducing our 2 characters. On the left is a pathogen. And when I say pathogen, it refers to any microorganism that causes disease. And on the right is our dear plant. And our plant is a castle. because it's a bastion of resources. It's full of sugars and nutrients gathered by photosynthesis and from the soil, and even from bartering with slightly nicer neighbors than our pathogen. And it is a safe place to replicate and an ideal host for our pathogen. And so we are going to accompany this pathogen as it breaks through the 3 layers of defense of the plant immune system, beginning with a moat, physical defense. These are the things that the plant does without even thinking about it. They're standing defenses. And a lot of them are easy for us to maybe think of off the top of our head or see and touch. Waxy coats on leaves help prevent bacteria and fungus from invading the leaf surface. Bark on trees is rough and tough. Thick cell walls, all these things deter invaders and act as this physical barrier between plant infection. Plants also produce toxic chemicals at low levels to deter invaders, but our pathogen has evolved to be very sneaky. Leaves have pores on them to allow for gas exchange, which are often exploited by invaders. They climb through the window, so to say. Some fungi can even apply immense pressure that can rupture the cell wall, forcing their way inside in sort of a cannonball approach. Pathogens develop tolerances for antimicrobials in the same way they would wear a gas mask to prevent being deterred by these early stages. So what I want to make clear is that the key to immunity is recognition. Keeping an immune system running on full defense mode is costly and not conducive to a happy plant. Our plant castle isn't fully armed and ready to fight at all times. It has built this wall and moat, but besides that, if it's not under attack, it wants to spend its resources on other things like growing and reproducing. Pathogen invaders will try to exploit this by sneaking in undetected for as long as they can. But once a pathogen is recognized, the immune system is swiftly deployed. And so the battle of immunity is really a battle of recognition. And there are 2 types of recognition I'm going to talk about today: general and specific pathogen recognition. Beginning with general, we're returning to our siege. The walls have fallen. The drawbridge is down. Our invader has secured a position inside the leaf and begins to attempt entry into the plant cell. However, our plant has prepared for this over courses of millions of years of evolution, and many invaders have similar signs of attack. Plant cells have receptors that recognize common signs of invasion, including bacterial-specific proteins that the plant wouldn't expect to be there, like a flagellin protein or just other bacteria-specific things. And cell wall damage can be a sign of a general pathogen attack. These patterns of invasion can be recognized and then turn on the plant defenses. However, pathogens have evolved ways to avoid this detection and are often able to elude the plant's recognition of common patterns. Specifically, moving on to specific pathogen recognition, our final layer of defense, in which case our invader has successfully pushed its proteins into the plant cell in an attempt to suppress the plant immune response so they can munch away at all the nutrients in peace. They are secreting proteins called effectors that are recognized by internal proteins in the plant cell called resistance genes. This is the forefront of the battle between plant and pathogen. It's specific diseases, specific bacteria and fungi that have corresponding specific receptors in the plant cell. The bacteria is actively trying to prevent the plant from recognizing it's there and activating its immune responses, while the plant is trying to come at it and recognize it and turn on those immune responses. So it's a battle of recognition, and it's an evasion and trying to find. The plant wants to recognize, the pathogen does not want to be recognized. So a lot of talk about recognition. What happens when recognition occurs? AKA the plant fights back. Once recognition is achieved, the defense system is launched. Hormones travel through the entire plant, sounding the alarm that there's an invader. Pores on the leaves will close to prevent further infection. The cell wall will thicken. The production of antibacterial and antifungal compounds ramps up. And in the most dramatic and effective display of defense, plant cells will die for the cause. Localized cell death at the infection site will starve the pathogen invaders and prevent replication and further colonization. It will cut our pathogen invader off from the supplies they came to seek And in this rice fungus I showed you before, it's cutting off the pathogen from any of the things it wants in an attempt to kind of starve it, kill it before it can colonize the rest of the plant. This is one of the more impressive ways in which we diverge from plants in an immune response. Plants are very regenerative and can afford to shed leaves and kill tissues to a certain extent before death, whereas we aren't going to lose a limb over a flu, if, at least I would hope not. So this is kind of an interesting way in which we diverge from plants. So back to our roadmap, we've done a quick tour of the physical defenses, the general and specific recognition, and then understood what happens when recognition occurs and the immune system is activated. And now we will dive deeper into the race for recognition, with our bacterial invaders developing new ways to avoid detection and our steadfast plants seeking them out. So I told you this was the forefront of recognition, with the internal proteins inside the plant cell trying to avoid and recognize plants, recognize they can set off the immune system. At the same time, the pathogen is trying to avoid detection, getting rid of the proteins the plant has. So this is where there's an arms race happening. The bacteria is running in one direction, the plant is chasing right after it. And I want to remind us that evolution is driven by natural selection acting on mutation. And evolution is what is allowing our bacterial foe to evade detection and our plant to recognize new attackers. And if evolution is driven by natural selection acting on mutation, is this a fair fight? Our bacteria can divide very quickly, sometimes as fast as once every 20 minutes. They can reproduce very quickly. They can colonize a plant leaf very, very fast. If some mutation or change in a gene occurs that helps our pathogen avoid detection, it will provide an advantage very quickly. Plants are much larger organisms, with lifespan at the shortest of a few days and the oldest, the several thousand-year-old redwood trees. So how does a beneficial mutation in a redwood tree that maybe allows it to recognize a new invader spread through the population? Even in less impressive but equally important plants like wheat or corn, the lifespan is a growing season. And so I'd like to argue that this isn't a fair fight. The bacteria are evolving much faster than the plants. However, in natural populations, plants are still winning a lot of the time. They're still recognizing these invaders and living healthy, happy lives. And so the central question is, how do plants develop new ways to recognize invaders with a slower evolution? Or more simply put, how do plants keep up? And this is the central part of my research, is how do these plants keep up with these and develop new recognition specificities? What we know so far is that natural populations act as reservoirs. In a natural field of wild grass, there's a lot of genetic variation at these recognition sites, in the same way that all the people on this Zoom call are very different from one another genetically, while we are all Homo sapiens. And so if a pathogen descends upon a field of wild grass, there's a higher likelihood that one of those slightly different plants will be able to recognize and activate resistance genes for it, in a very classic evolution example of natural selection. A newer discovery is that the plant's tools for recognition, their receptors, are evolving faster than their other genes. And this is because some genes mutate faster than others. And that's due to the nature of the position of the gene in the genome and on the chromosome, how much the gene is expressed, and any decoration on the DNA. There are different mutation rates across our entire genes. And also, some genes, it's more important for them to be variable, while others, it's more important for them to stay the same. What we don't yet understand is that if this standing population variance and the higher mutation rate is sufficient to generate enough diversity for these plants to survive and come up with keep up with their pathogen invaders. And I'd like to think for a second about our own immune system. When you get a vaccine, you're able to generate antibodies to bind to whatever protein has been injected, which I'm sure we're all intimately familiar with the current pandemic and discussion about vaccines. A vaccine wouldn't work in plants in the way we understand them. There's no active antibody generation. But I think potentially there is a more active mechanism of mutation in plants to generate this new recognition at a higher rate than we previously thought. And so this is the real forefront. This is the hot button question of how are plants keeping up with their slower evolution times? Are there other factors at play than what we already understand? So looking back with our fabulous knowledge about the plant immune system, we then looked at how plants are recognizing new pathogens in this continuous race for recognition, with pathogens evolving evasion and plants evolving recognition. We looked at the speed of evolution and saw a distinct bacterial advantage, but hope was not lost. Plants are very successful, but that brings us to the question of how do plants keep up? And is there an active mutation mechanism that's kind of aiding this evolution of new recognition and keeping them on par with the things that are invading them? I want to leave you with our rice infected with fungus, because I want to remind you that we're not bystanders in this siege. We are strongly allied with plants because of our reliance on them for food, fuel, and medicine, and therefore we are very invested in their health. Plants have effective, functional, and layered immune systems that are remarkable at protecting themselves, and they evolve recognition specificity faster than you would expect. But there is still so much we don't understand about how plants defend themselves against their diseases. And it is imperative for our future food security for us to fight against plant disease. And to do that, we have to understand how plants defend themselves.
Chandler, great, thank you. I warned Chandler before beginning, for opening the waiting room, that I had some very simple, biologically simple-minded questions, and I will let one of those rip if there's not a question from the audience here, from the gallery. Would anyone like to, here we go. Stuart, I believe it's you. Would you like to ask the question yourself, or would you prefer that I read it?
Sure, I'll go ahead. Thank you, Chandler, for your presentation. I enjoyed it. You got so many questions, including about photosynthesis, but I won't ask that one now because I have limited time. With regard to your how do plants keep up, I'm curious about the development of plant immunity, either by their own propagation, through flowers and seeds, or by human-directed hybridization. How is this different with regard to plants that do not propagate by flowers, like bananas, for example? I heard that there was a terrible fungus in Central America about 80 years ago or so. So I was wondering how that is handled. Do you just throw out the old trees that are getting attacked because that variety is not strong and replace them, or do you somehow help the existing crop, the existing farm of banana trees, or other trees, whatever? Thank you.
Thank you for the question, Stuart. This is definitely a very active area of plant research, and part of why it's so exciting to study plant immunity is that when you do have a situation like a fungus attacking a tree, like how do you engineer a new resistance gene and breed it into a population like a banana population? In terms of your question specifically about things not propagated by flowers and seeds, immunity in like clonal propagation, things like bananas, is really interesting because they're not generating that diversity and kind of new recognition specificity like you would think a flowering plant would. Any sort of flowering, like sexual recombination, is going to introduce a lot of diversity, which is great for immunity. And so when it's something like banana, it's like, what's going on? So still a very active area of research in terms of how are they producing sufficient diversity to find new recognition. And I think part of that points to the question of, is there a more active mutation mechanism going on? In terms of throwing out the weak and susceptible trees, I think at our current level, it would be, yeah, hopefully there's some wild variety that has a resistance that we can help breed into the population. But the hope is to be able to engineer these new resistant strains without sacrificing things like deliciousness and yield of bananas that we've worked so hard to select for.
Thank you. Mhm.
Thanks for the question, Stuart. All right, if there's no one else to ask, then Chandler, I get to let one go here. When you showed the pathogen, see when you symbolized it in your drawing there, it was interesting that you chose something that looked like, to me anyway, like a COVID virus. I wonder if SARS-CoV-2, I guess that's the right name, is it true that attacking proteins, say, or whatever virus may do, is that a plant pathogen really would, to a casual observer, look like a pathogen for humans or animals at least?
Definitely. Viruses attack plants the same way that they attack us. And there are some bacteria that attack both plants and humans. And a lot of like foodborne illnesses are because they're soil pathogens that, haven't been washed off or removed otherwise in the food system process. And so a lot of them look very similar. And that's why I think plant immunity is so interesting, and studying it can also inform our own immune system and kind of disease susceptibility, because in the broad tree of life, we are related to plants, somewhat distantly. There's a, well quoted fact that we share 30% of our DNA with bananas. And that's not entirely true. We're very different from bananas, but there are things we can learn about our own immunity by studying plants. We all share the same kind of bare bones. So yeah, there are viruses that attack plants that look like SARS-CoV-2. And I picked it to kind of inspire. We're living in a large, pandemic sphere right now, and sometimes it's nice to focus on another species' diseases and problems for a while.
Great, thank you. All right, I have one more that's so basic I'm embarrassed, but if anybody's holding back because they think their question's too simple, just give a listen to this one. I understand that plants have a special talent, and even in regard, let's say, to the entire realm of living creatures on Earth, and that is photosynthesis. And animals seem to have a rather special gift in their mobility. How far do we have to go back to try to understand historically, to understand why it is that there aren't, except for the Venus flytrap, the few exceptional plants that move a little bit, how is it that those two skills, mobility and photosynthesis, don't, as far as I know, coexist on a creature on Earth?
Okay, I will answer your question, but I will first give you an example of where it does exist, kind of. There's a type of sea slug that has a very tight coexistence with an algae, to the point where they're starting to get to that line of, are they separate or not? And so the sea slug will photosynthesize, kind of. It's an algae on top of the sea slug that's so tightly interacting. There's debate on whether or not they're still independent organisms, or if they're a photosynthesizing sea slug. So there are some very fringe examples of mobility and photosynthesis. But to answer your question more directly, photosynthesis became a trait of multicellular organisms like plants in a very ancient endosymbiotic event. So that's one cell engulfing another and then very slowly becoming, very deeply intertwined. It's why eukaryotic cells have things like mitochondria and chloroplasts where the photosynthesis happens. It was a very ancient engulfing of cyanobacteria, which is the ancient photosynthesizer. And so I think that happened after the split into animals and mobile creatures versus not. And algae do swim around, so there is some mobility, but I think probably it just became an evolutionary trade-off. It's better to not waste energy on being mobile and just stay in one place. But it does provide a lot of interesting challenges to be rooted to one spot.
My wife is not sympathetic to my argument about why I don't get out more. I am just sitting on the sofa, not wasting energy. Yeah, that doesn't apply.
I'm not wasting energy.
Chandler, thank you. Thanks very much. I don't see any more questions, so you're off the
I
hook.
think I do have one more question.
Oh, good. Where do you see it?
It's in the chat.
Ah, good.
Yeah, so
Stuart, yeah.
okay, yeah. So seems to me that we assume that there's lots of pathogens in the soil. Dogs poop on the soil or in our gardens, that sort of thing. So we wash off leaves and veggies and fruits before we eat them. So how is it that those scary, dangerous pathogens aren't inside the fruits and the leaves and the veggies that we eat? And if they are, and maybe they are, and if so, would it be best to always cook them? Thanks.
That's a great question. I think they definitely can be in the foods too, but I would say that most dangerous, scary pathogens are soil related because you're absolutely right, of like feces and kind of manure type things. Most of the pathogens that attack plants are going to have we won't have any issue with. And I like to think about it in the same way that like every surface you could imagine is completely covered in microbes, and including our intestinal tract. And our intestinal tract is also very good at keeping us from getting sick. So it's kind of it's a scale issue. Like if there was one horrible pathogen on the plant that like you rinse it off, and then by the time you eat it, by the time you digest it, it's very unlikely that it's going to get you sick unless it's like in a high quantity. So I don't think we have to cook all foods. I love raw fruits and vegetables, but it would be safer. Maybe in the same way it'd be safer to wear a helmet all the time.
So does that mean that the skin outside of the leaves and the fruits and veggies is effective in keeping those pathogens out? Like salmonella, listeria, things that would get us sick.
I would say yes, like plants are effective at keeping things that are bad for the plants out. And I would say things like salmonella and listeria don't typically infect plants. It's in situations where there's a lot of fertilization, like kind of more factory farming style, like very high fertilization with like manure, that you get enough salmonella on like romaine lettuce to have it recalled. And so I would say that, yes, it is safer to cook these things because then it would destroy them. But I would say not necessarily that like salmonella would be colonizing a strawberry in like a more natural setting.
Okay, so it's on the outside, not inside. And when you mentioned our gut bacteria, I think you were talking about, are you saying that a healthy, varied population of gut bacteria is effective at finding and fighting and killing off these pathogens when we do ingest them? Is that what you were referring to?
Our gut microbiome, and even the microbiome of plants, is a great defense against things trying, like pathogens, because if there's a varied and distinct microbiome, then the pathogen not only is trying to get into, and fight us, it has to compete with all of the other microbes that are there. So healthy microbiomes are really important for us and for plants.
Cool, thank you.
Thanks, Stuart. I see Demon Druid has a question there. Demon, do you want to ask it yourself, or would you like me to read it? I will take your silence to indicate I should read it. Here we go, Chandler. I have heard, Demon Druid has heard, that humans are more not human when we include all our necessary other living things. Is it similar for plants? Do they have microbiomes or anything similar to that kind of helpful inside life?
Okay, yes, I understand the question. Yes, plants I talked about pathogens today, but just like us, plants are covered with beneficial microbiome, especially at the root level, to help import nutrients from the soil. And I think this gets to your question a little bit, Stuart, too. There are fungi that live within plants that are strictly beneficial. They're called mycorrhizal fungi, and they live within the plant cells. And there's this intense bartering system of nutrients going back and forth in this kind of mutualistic reaction. So yes, plants have root microbiomes, leaf microbiomes, and fungi living within them that are all beneficial or at least neutral to not be classified as pathogens.
Thank you for the question, Demon. I think I've never said that sentence before. Chandler, thank you. We'll have a chance to give Chandler a big round of applause at the end of the entire Ask a Science Envoy adventure here. But for now, let me introduce our next speaker and ask him to share his screen. Stanford astrophysicist Alex Madurowicz will present Imaging Extrasolar Planets. Alex earned his bachelor's degree in physics at UC Berkeley, where he was awarded the physics department's Olson Scholarship and was a research assistant at the Lawrence Berkeley National Lab. Now he's a fifth-year PhD student at Stanford. Please welcome astrophysicist and Wonderfest Science Envoy, Alex Madurowicz.
Thank you for that introduction, Tucker. So it's going to be a bit of change of pace. We've been firmly rooted to the ground, but now we shall lift off into space. So I'll just get started by asking you to imagine, or rather just look at your screen and visualize looking at the sky through a turbulent fluid. You can sort of very vaguely make out there's a sun and there's maybe some clouds, but it's hard to see what you're looking at because of the distortions that are introduced. The image is blurred and messed up by turbulence in this fluid. Now, right now, we're actually stuck at the bottom of a pool, which is not exactly the best place to build a telescope. And you might notice, if you look at a pool, you can see these patterns that form on the bottom from the light being bent by the surface. And as you step down deeper into the pool, it gets more distorted and more bent. And so when you're building a telescope, you don't want to build your telescope at the bottom of the pool. You want to get as close to the top of the pool as you can. Now, the pool is a metaphor. The actual turbulent fluid that matters for astronomy is the atmosphere, the air that's above us that we breathe. And so astronomers build telescopes on the very, very tops of mountains. This picture is of Mauna Kea, the volcano on the big island of Hawaii, taken from the window scene of a plane at like 10,000 feet. So you can see all the way from the ocean level at the bottom to the very top where it's even like covered in snow. And if you look closely, you'll notice a couple of little tiny specks. These tiny specks are giant industrial buildings, like something like 4 stories tall, that contain the largest mechanical eyes that humans have built. These telescopes are giant. They're tens of meters across for their primary mirror, and the whole thing is encapsulated in these giant domes, and they're really quite marvelous. There's a picture of me standing next to one of them. And so when you look at the sky using a telescope, this is a video that was taken with a rather ordinary telescope, one that a hobbyist might have. You can begin to make out these distortions that come from the turbulence in the air. I sort of have 2 examples of different lunar craters because that's just something you might look at it with a normal hobbyist telescope. But this distortion matters a lot when you're looking for extrasolar planets. And so what do we do about this? Well, we have to fix it. And the way that we fix this problem is by means of a machine that unbends the light. The technology is called a deformable mirror, and it's a piece of metal or glass, metal and glass layered together. And behind it are a bunch of little tiny pistons that can push the sheet of the mirror into an arbitrary shape. And the instruments which are deployed on the telescopes rapidly change the shapes of these pistons every millisecond to fix the fluctuations in the distortions of the atmosphere, to unbend the light and make the image sharp again. It's really quite a modern miracle, but that's not enough. You can't just fix the atmosphere. You also have to block out the starlight if you want to see a planet. This was a problem that was solved in the early 20th century by French astrophysicist Bernard Liot. There's a picture of him at the French Academy of Science. where he discovered that if you want to block out the starlight, and in his case, he was thinking how to block the starlight from the sun because he wanted to create a solar eclipse inside of his telescope. That it's not just enough to block the starlight right here with one mask. You need a second one, and you need a second lens, and you need to block the outside of the beam. And the reason for this is because of wave optics, which is all what we study in physics. And sort of rough idea of what's going on here, you bring the light to a focus, and you have this mask, which is basically an opaque circle in the center, which you block the light from the focus, but it doesn't block everything. Some of the light gets pushed out to the side, and that's by design because you want the majority of the light to go out to the side of the beam so that this circular washer-shaped second piece, which is absolutely critical, can then block the majority of the starlight. This was the invention of Bernard Liot, and it is called a coronagraph because it's used to study the corona of the sun like you would during a total solar eclipse. This photograph of the total solar eclipse I took with an ordinary camera when I traveled to Oregon in the summer of 2017 to watch it. And I honestly could not recommend the experience anymore. It was truly an amazing sight to witness. And the good news for you is that there's another one. If you haven't had a chance to see a total solar eclipse and you want to, April 8th, 2024 in America, there's another one happening. And I would say go for it. I'm definitely going. There's really, really no astronomical experience like seeing the moon completely block the light from the sun. So you got to go. It's really life-changing. I would recommend it more than anything else, more than the northern lights, more than a meteor shower. All of these things are peanuts in comparison. And just for fun, there's another picture of me with my pal Max in Oregon wearing our goggles. So if you're going to go watch the total solar eclipse, make sure you get a pair of these glasses, which let you look at the sun directly in the moments leading up to the eclipse. And then during the eclipse, you can take them off and put them on back when it's over because you want to protect your eyes. So with that aside, out of the way, back to planets. With this technology, we can deploy and fix the air, make the light back into a perfect beam and block the light of the star. And what do we see? We see planets in orbit dancing around their host stars. And these videos are actual data. These observations were taken with the Gemini Planet Imager, which is the instrument that my advisor built over the course of his scientific career. And they're like, these really make my heart fuzzy. You know, just looking at this, what we're seeing are planets orbiting their own stars in their own systems, very, very, very far away. And it's truly a marvel of technology that we're able to mix this. You might notice there's a lot of sort of bright, noisy things in the center next to the mask. That's where the coronagraph is. This is sort of residual light that is an imperfection that is because our machines aren't perfect. They're delayed by a few milliseconds, so some light gets scattered into the images. And these images are heavily post-processed and combined in order to maximize the signal. And so there's still a long way to go. These planets are absolutely giant. These are massive planets. They are very bright. And in fact, they're so bright that there's no planet like how bright these planets are in our own solar system. But I will have a somewhat decent analogy a bit later. Something else that we can do with this data is split the light from the planet into its different colors. And so in this animation, we're stepping through a bunch of different colors in the near infrared, which is just beyond the red spectrum of light. And you can notice that there's this peculiar feature. It gets brighter and then dimmer around 1.5 microns. And this absorption feature is characteristic of methane. And so with these observations, we can infer the composition of the atmospheres in these planets and start to infer about the dynamics and the processes which shape their atmospheres as well. But what kind of planet are we looking at really? Is there an analogy in our solar system that may be more familiar to the person? These planets are just a speck. So I have this really nice video of Voyager 1 on its way out of the solar system approaching Jupiter, which is probably the best example of a planet in our solar system that is like these other planets because it's just so big. It's, you know, Jupiter is the king of the planets. It's absolutely massive. And its atmosphere is deadly, toxic, and dominated by this absolutely gargantuan cyclone, the Great Red Spot, which you can see swirling around. This hurricane, essentially, on the planet has been there for at least 300 years, and it shows no sign of stopping. So you really wouldn't want to have to fly around or live inside of it. It would be quite a messy place to be. And so because Jupiter is so close to us and so big, there are so many great observations of Jupiter, like this picture, which was taken by the Gemini Observatory, and it shows Jupiter in the infrared. And so you can see all kinds of interesting things in this picture. There are very different kinds of cloud layers of ammonia that are at high altitudes, and these are the dark bands where there's less infrared light or heat coming from it. And then there are gaps in the clouds where you can see deeper into the vortex of the planet. And these are the places where it's glowing bright orange from all of the thermal energy escaping from the deep interior of the planet. So a planet in the infrared looks like this, and the planets we can see in the other systems are probably similar to Jupiter in this way. And so you can use this picture of Jupiter to imagine what these planets are like. But if you want to know a little bit more about thermal imaging, there's a fun demo that we used to do at the SLAP Community Day, which is a public outreach event here in the Bay Area. I think they might actually be having one this year now that the pandemic is sort of easing its way. I made this little app to take pictures with this thermal IR camera that you can just plug into your phone. And then that's a photograph of me and the ex-Stanford soccer goalie, Andrew Thomas, and Kieran Gilmore, who did a rotation project in our research group, as well as some other people interested in science in the background. And so what you're looking at is just the heat coming off of our bodies. And you notice I'm wearing some sunglasses, and so those block the heat coming from my eyes. So I've told you about planets, how we can see them right now using these wonderful technologies, but how might we see more detail and smaller planets? What kind of things might the future hold? One of my favorite ideas is the possibility of creating an artificial eclipse outside the telescope. In this NASA animation, what you're looking at is the deployment of a star shade, a petal-shaped occulting wedge that you deploy from your telescope and send off to a great distance to block the light from a star you want to observe the planets around. Now, this star shade has a very special shape. It looks like a flower, and these tapered edges are extremely important. Believe it or not, if you just had a circular obstruction to block the starlight, the light would diffract or bend because it's a wave around the edge and interfere in the very center, producing this bright spot, which was, believe it or not, the center of a famous physics debate in the 19th century. And so the star shade has been designed to fix this problem by bending the light the other way so that it stays out of the center. And so you can create a really deep shadow, which you can position your telescope in to have a very natural observation window to see these faint planets. Now, I love the star shade because the physics is really wonderful, but I hope you agree with me in just looking at the deep logarithmic shadow that's formed from this petal-shaped occulter is really, truly just beautiful on its own. I really love the fractals that this forms. And it's not just pretty, and it's not just fancy, pictures and whatever. Real scientists have studied this idea extensively and have proposed a concept which could revolutionize science in the coming decades. This is a simulated observation from the HABEX final study which was a proposed NASA mission concept to deploy a star shade along with the telescope to directly image entire systems of planets all in one go. So you could get spectral snapshots of entire systems of planets and infer the composition of the atmospheres of all of the planets in a single snapshot in an entire system. And you would be sensitive enough to even find planets that are small like Earth. But unfortunately, this concept was not recommended by the Decadal Survey, which is one of the major bureaucratic endeavors that the astrophysics community engages in once a decade to sort of decide the future of the field. And instead, the recommendation was to focus more on coronagraphs because of the problems associated with navigating the star shade and the telescope into position, the sort of a fuel cost associated with maintaining alignment that makes looking at a large number of planets really difficult. But it's still an interesting concept. And I think it's still pretty neat to show. Now, going even further into the future, this is a concept that really blows my mind. What would happen if you want to try and actually resolve these planets? Like you wanted to see the details on them. You wanted to see the surfaces and their atmospheres, like the blue marble image of Earth, where you can tell there's Australia and there's Japan, and look at it's, you know, there's the jet stream that's, you know, the winds and the high altitudes and all of the clouds and all of the beautiful things you can see on the surfaces of the planet. What would it take? Is such a thing even possible? It's extremely difficult. And I'm going to try and convey to you how absolutely ridiculously difficult creating an image that's this detailed of an exoplanet would be. And to do this, we need to first understand the size scale of these planets. They are far away and they are small. You might be familiar with seeing the moon. It's about half a degree, so it's like the size of your thumb from arm's length. And you can see it with your eyes, it's no problem. But how big is a planet? Let's just zoom in really quick. We're stopping in the outer solar system at Pluto, which is the furthest planet you're probably familiar with. It's very tiny. Zooming in even more, the famous M87 image of the black hole, which was taken by an Earth-sized array of telescopes. And the planet is much, much smaller than that. The image of M87 is the highest resolution image ever produced, and we want to go maybe 100 or 1,000 times smaller. How can it even be possible? If you want an ordinary telescope to do this, it has to be gargantuan, literally the size of the sun. This thing would qualify as a megastructure, like a Dyson sphere. There's no way that you could get Congress to fund a telescope that's 250,000 kilometers in diameter. It's ridiculous. It's out of the question. But we can't give up hope. There are some clever people out there with some smart ideas. When Albert Einstein invented general relativity, one of the major consequences was gravitational lensing. This simulated image is of a black hole. I hope you know what a black hole is. It's an extremely dense astrophysical object, which is so dense that light cannot even escape. And it distorts space-time around it and bends the light in this really peculiar pattern. And you might not really be familiar with this kind of a lens. It's in fact kind of a strange kind of lens, but there is an example of something that you might have in your house right now that lenses just like this gravitational lens. And it's the bottom of a wine glass, believe it or not. So I made this little video with my laptop and, you know, moving the wine glass back and forth, but it turns out the base of the wine glass lenses light in almost exactly the same way as a gravitational lens does. And you can see all kinds of things. There's the two images moving sort of in opposite directions. You can see the formation of the Einstein ring when it passes in front of the bright star. And so if you want to, you know, show a demo to perhaps young and interested family member or friend, this is a great demo to get them interested in gravitational lensing. But what good is a gravitational lens to us? Is there something we can use that would be a component of a telescope? There is, and it's the sun. If you have the gravitational lens of the sun, you might be able to do it. The way the problem is kind of like this. You want to look at a planet. The sun lenses light, just like the black hole, but weaker because it's not as massive. And it bends this light into a focused image, but the image is all messed up because it's a pretty crappy lens. I mean, you never want to look through the bottom of a wine glass. So you have to find a way to fix this. And one of the existing ideas out there was you send just a bunch of telescopes to peruse this image, and you make a bunch of measurements and you can make a reconstruction. But the topic of my research, and in a paper that was quite recently accepted, literally last week, we came up with a new method, a way to do this better. So by observing with the gravitational lens of the sun, it turns out that the ring that forms is not perfect. It's a mixed up image of the planet. And by undoing the mixing up with some fancy math tricks, you can reconstruct low resolution images of the planet from these single observations with the solar gravitational lens. But there's a whole bunch of caveats, and I'd be happy to answer all of the questions of caveats in this section, but I'll just leave you with this idea that you could maybe actually see the surfaces of planets by using the gravitational lens of the sun. And I hope you enjoyed that. I think that my time has certainly ran out. So that's all I have to share. Thank you so much.
Wow, thank you, Alex. Man, let's see. We've got some questions. Let me move the chat up here. Please don't be shy about you get some good applause. You'll both of our great speakers will at the end here. I'll fire a question off. I want to ask about the image you showed early on, Alex. I think it was the methane image. Was methane from one of the planets orbiting 51 Pegasi?
Yeah, so it's 51 Eridani.
Sorry, Eridani.
Yes, it has a lot of methane. So it's quite the smelly place.
And we're sure that's not methane, forgive me, in the atmosphere of the star. It's in a planet, huh? Because we see that the occultation is of the star, but we're confident in the light that's getting to us, that's glimmering there around the blocked star. That light contains the spectrum of methane, and we're confident that's coming from the planet.
Absolutely. So there obviously is some contamination from the starlight. As you can see, there's quite a bit of extra starlight leaking around the edge. So this can be calibrated. You can make very precise measurements of this color of the starlight, and you know exactly how much starlight is at this spot, because you can just say it's about the average of the amount of starlight at all of these other spots where the planet isn't. And so you can very carefully remove the contribution of the starlight to this point and know that this signal is coming from the planet.
Wow, that is marvelous. All right, I get another question. The gallery is too. Oh, no, we have a new message. Let me scoot down there. Stuart, do you want to ask it yourself?
Sure. Thank you, Alex. Thank you, Tucker. Alex, during your presentation, you were kind of talking a little bit about other telescopes, and so I'm not sure if you covered this. The new James Webb, I presume that this imaging process has not been incorporated into that new space telescope. So is there any way to incorporate that in the images that it is going to be taking soon?
Certainly. So James Webb as a space telescope doesn't have to look through the atmosphere. But believe it or not, it does employ a very similar technique to the deformable mirrors. Because James Webb folds up to fit inside the rocket, when they deploy the mirrors, they have a bunch of segments, a bunch of pieces, and they need to align these segments to create what they call a phased array, so that all of the segments act together as one giant mirror. And the process of doing that and moving the segments back and forth and trying to get them to align perfectly is very similar to the problem of fixing the atmosphere with a deformable mirror, except it's a lot slower because you don't have to rapidly do this as the atmosphere is changing. You can take as much time as you want to look at a star and make very slow changes to see them align and calibrate all the mirrors to make a perfect point source. And so they use very similar techniques when they're calibrating the James Webb Space Telescope.
Okay, so you're saying that they can deform the mirrors in real time or whatever while they're taking pictures to account for.
It's sort of like in loop with the ground. So they'll take a calibration image. They'll say, oh, this segment needs to be shifted over a little bit. And so on the ground, they're going to do some calculations, and then they'll send the commands up and they'll move the mirror a little bit, and they'll take another picture and they say, oh, now we have to move this other segment a little bit. And so there's this iterative process where they slowly calibrate the exact positioning of all of the segments to make them into one mirror.
Mm-hmm. Okay. But with regard to your filtering process to filter out the light of the stars so that you can see the planets.
Yes.
That's what I'm really asking about as far as.
Oh, I see. So the coronagraphs.
Yeah.
So Webb does have coronagraphs, but the coronagraphs are old. Webb was designed like 20 years ago, and the coronagraphs that we could design 20 years ago were not so good. And so it turns out that Webb is not the best at looking at planets. Webb really shines well when it's looking at faint, distant galaxies, which is what it does best. You can do this same technique where you block the starlight with Webb, but it's not better than the telescopes that we have on the ground, which are much bigger and do fix the atmosphere almost perfectly. But Webb will do really well at wide planets. So planets that are really far away from their stars, that will be somewhere that Webb will be really successful for planet imaging.
Great, thank you.
Yeah, thanks, Alex. Thank you, Stuart. All right, I'm going to fire another question here. Alex, you used a verb that I haven't seen associated with general relativity in the actually many talks and a few books that I've read on the subject. And that is, I think you said that Einstein invented general relativity. Now we're tiptoeing here into philosophy, but this is Wonderfest after all, so why not? Help us understand why you chose that verb. In what sense did Einstein invent, as opposed to, say, discover, general relativity?
Might not be the appropriate verb. So I think that was just me coming up with something on the spot, and it was not intentional.
I love it, though, because it really made me think about just what was Einstein's contribution. Clearly, he in a sense, yes, he'd used somebody else's mathematics, but by joining that mathematics to describe the curvature of space-time, oh my goblin, maybe there's a lot of invention going on there as opposed to discovery.
I think there is a fine line between the two. On the one hand, discovery sort of emphasizes that it's already out there, just waiting to be found. And then the invention, on the other hand, emphasizes the human experience of exploring and finding and creating, which you kind of have to make the connections between ideas that will let you make some new realizations like this.
Yeah, I think you just paid Einstein a very high compliment, and I applaud you for it. I see no more questions, so let's bring this event to a close. I want to thank everyone in our gallery here for showing up. Sometimes I refer to Wonderfest visitors as Wondernauts, and if you're not embarrassed by that term, please accept it. Thank you for being curious and being here. Especially to those of you who are Wonderfest supporters. There are donors and folks that are called patrons who help fund Wonderfest through Patreon. Thanks to you, of course.